Okay, guys, I guess this is my second attempt to record this chapter. My first one, I got disconnected because of, I guess, internet connection. So I hope, let's go over this quickly. So this chapter is about how cells divide. One cell becomes two cells. Of course, the chromosomes divide and the process of meiosis. You do have labs that happens in the ovaries and testes, and the process of meiosis. Let's talk about that. So chromosomes are made up of DNA and protein molecules. So collectively, the DNA and protein is called chromatin, but when they condense and become visible on the microscope, then they're called chromosomes. How many pairs do we have? We have 23 pairs. So on your quizzes, make sure you pay attention when you're reading your quiz or your exam. Look at the term pair, or it does not say a pair. We have 46 chromosomes or 23 pairs. 20 out of those 46 chromosomes, 22 of them are autosomal chromosomes, autosomes, and only one pair is sex chromosomes. The sex chromosomes for male is XY, and for female is XX, which we talked about it in last chapter as RY gene. And so when they make a karyotype is when they set up the chromosomes in a way so you can see them on the microscope nice and neatly right here, and they align them that they arrange them from the largest chromosome to the smallest chromosome. Okay, so that's chromosome number 1, number 2, number 3, number 4, number 5, number 6, number 7, and so on and so forth. And the very, very, very last one is sex chromosomes. And the one to the very last sex chromosomes is the smallest chromosomes, are chromosome number 20, autosome chromosomes 22. So mitosis is the division of the cell. When you cut your finger, like me, I cut my hand here, that is going under mitosis. Right now, the cells in here are undergoing mitosis. When you have a little kid growing at home, it's going under mitosis. When you have a plant growing and becoming taller, that plant is going under mitosis. Okay, but what happens in the ovaries and testes, it is called meiosis. There is no T. So what happened? The chromosomes have to duplicate. They have to become 2. And what happens when you have one chromosome becomes 2, right? And then each one of these will go to a separate cell eventually, okay? So now this is also called a chromosome. This is a chromosome. This is also called a chromosome. But this arm right here, I'm drawing it, that's called the sister chromatid, okay? And the other one is also called the sister chromatid. So this is a sister chromatid, this is a sister chromatid. And they are connected together right here where my elbow is. They're connected together by centromere. It will come up, okay? So here we go, centromere. That's where the two sister chromatid are attached to each other. Still one chromosome. This is one chromosome, have not been duplicated. This is one chromosome, have been duplicated, and they're attached in my elbow at the sister, at the centromere, which is called, each sister chromatids are attached. Okay, the cell cycle. So what happens, Amir, when one cell becomes 2 cells, and then 2 cells eventually become 4 cells, what happens? Then we should become, if we are 1 trillion cells, then soon you become 2 trillion cells. No, cells do die. When the cells undergo mitosis, some of the cells die. They're programmed cell death, which is called apoptosis, which will come up. Okay, so anyhow, the cell cycle has 2 parts, the interphase and cell division, which is we called mitosis. Okay, and then when the cell is not undergoing the division, the chromatin appears to be a tangled mass of thin threads. Yes. So interphase, there are 3 stages into interphase. Interphase is not stage of mitosis, and I will talk about mitosis. The three stages of stages of interphase are G1, it means growth, S, synthesis, it means your chromosomes duplicate, and G2, the second growth of the cell. Here they are, growth and DNA replication. Right here, that's the S, growth, and then growth and final preparation for division, and that is called the G2. G1, S, and G2, and those are the stages. The interphase, again, very important. Our nerve cells, our neuron and muscle cells, they do not divide. You were born, that concept, I think I talked about it, it's called utility. You were born with a set number of neurons and muscle cells, and you will die. I will die. I will speak of myself. I was born with, let's say, 1 million neuron and 2 million muscle cells, and I will die. If everything goes normal in my life, I do not come across Alzheimer's or anything else, muscular dystrophy or anything like that, I should die with 1 million neuron, and I should die with 2 million muscle cells. Okay, I made up those numbers. That concept in biology is called utility, constant number of cells. So nerve cells and muscle cells do not undergo mitosis, which is recently they're discovering that's not true every 7 years. That's what, based on what I'm reading, every 7 years, your whole entire body dies, the cells in your body dies, and new cells grow. But they do not undergo the normal routine of mitosis as we know. So the cells get into a G0 stage. Again, we can talk about that for a while, but let's just move on. Hopefully you become interested in science and you study that stuff a little bit more. So mitosis, the M stage, M is for the mitosis and cytokinesis is where the cytoplasm divides. Mitosis is typically under division, duplication, and cytokinesis, division of the cytoplasm. This is what I was talking about, apoptosis. Apoptosis, it means programmed cell death. So when one cell becomes 2 cells, eventually these cells undergo mitosis. You cannot have 1 trillion cells cannot become 2 trillion. So some of the cells die, and that is programmed cell death, which is called, that is different than necrosis. You probably have heard of that, necrosis. That is, it caused from injuries and problems that occurs. That's different than apoptosis. So when the cells are dividing, they are checkpoints. The cells cannot keep multiplying and multiplying and multiplying. That's what happens with cancer cells. They escape these checkpoints. Okay, they do not respect these checkpoints and they keep multiplying. Right here, they are, your textbook is talking about these checkpoints at G1 and G2 and M stage, and talks about it a little bit. I'm not going to talk about it, so you guys make sure you read it and you understand it a little bit. Here is G1 checkpoints, and then continued P53. It is very important protein molecule. I guess we talk about this one again when we talk about the cancer chapter. It can stop the cell cycle, yes, P53, and then that's the scientists are trying to work with that molecule and make that up in case of cancer patients. But anyhow, the cell cycle halts and undergoes cell verifies that the DNA replicates and so on. G2 checkpoints, okay. Mitotic checkpoints, okay. External controls, what happens, external signals comes from signals transduction pathway. Here it is, the adjacent cells send a signal right here and tell the cells to stop growing, and these are the sequence of events occurs. External controls discontinue. Very important. Expression of, when we get to cancer chapter, this will come back to these terminologies. Might as well you get to know it a little bit here right now. The expression of these genes may stimulate or inhibit the cell cycle. So genes called proto-oncogene, oncology, you've heard of that term. It means study of cancer on. Okay, study of cancer. So proto-oncogenes stimulates the cell cycle, and the genes called tumor suppressor genes inhibit the cell cycle. We'll talk about this when we get to cancer chapter, but these are called proto-oncogenes. Oncogenes are a set of genes that are involved with cell cycle and so on and so forth, which we'll talk about it later on. Okay, mitosis. This next few slides are going to go over the stages of mitosis. Here, as I talked about, if you have a cut skin, right here, you must see my band-aid. The cells undergo mitosis, and of course, when zygote, last chapter, zygote becomes two cells become four cells. That's all mitosis. So, what is mitosis? Let's go over mitosis quickly. Here is a chromosome, and then, as I said, this is a sister chromatid. This is a sister chromatid, and they're attached together at centromeres. Okay, and here they are. They're showing you the state, what happens. So if a cell has 4 chromosomes, 2 blue and 2 red, at the end, you will have with 2 blue and 2 red. Okay, so 2N, it is diploid. You've heard of that term before, diploid cell, and you end up with diploid cell. You started out with 1 cell, you end up with 2 cells. Okay, that's mitosis. Centromeres, the centrosomes, centrosomes, they are microtubules which pull the chromosomes apart. And I guess mitotic spindles, aster, they are array of microtubules, and then again, you will see that these are the stages of mitosis. What happens during prophase, right here, these are real pictures, these are real pictures, these are cartoons. What happens, these are the centrosomes right here, and these are microtubules, and they attach to the chromosomes and pull them apart. So, and the asters, these are asters right here, they're microtubules. And what happens, the very first stage, do they mention them in here? Prophase, metaphase, anaphase, telophase. It does not. Pre-prophase, metaphase, when the chromosomes line up in the middle of the cell, all of them line up right here. That was, you know, these are all prophases, and right here, metaphase, when the chromosomes line up in the middle of the cell, right here, you can see that. Anaphase, that's when the sister chromatids separate. Okay, so the sister chromatids separate, and they're V-shaped looking right here. Look at that V-shape. They're V-shaped looking, and that's sister chromatid separate. And then when the sister chromatid separates, now the cleavage furrow right here, you can see that's telophase, and you can see the cleavage furrow. So anaphase is talking about what happens during anaphase, telophase, everything. Cytokinesis, that is when cleavage furrow is occurring. Here is a cell membrane. They're going inward, and then now you have 2 cells. But cytokinesis, it is right now, I'm dividing the cytoplasm, this is cytoplasm. I'm dividing the cytoplasm itself too. So what is meiosis? That was everything I talked about so far was mitosis. What is meiosis? Meiosis only and only occur in ovaries and testes. That's it. Nowhere else in your body occurs. And then at the end, you make a haploid cell. I have 46 chromosomes. In my sperm, there are going to be only 23. In your eggs are going to be 23 chromosomes. Okay, I hope I'm making some sense. Meiosis introduces genetic variations. Of course, sexual reproduction is always in biology. It creates genetic variations, and it is very important in order to survive. Then members of a pairs of homologous chromosomes or homologous to chromosomes are homologous. So this chromosome, for example, is orange color. This chromosome is orange color also. And they're wandering around inside of the cell. They're moving around inside of the cell. But when the order comes that you need to divide, they come together. That's what is called homologous. You saw that in karyotype at the beginning, there were 2 chromosomes of number 1, and they were homologous chromosomes of number 1. So right here is a tetrad. When the homologous chromosomes come together, it's called tetrad. And right here, the blue are together, the size, of course, the small ones and the large ones are together, red and blue and red, and each one of them in meiosis 1. So a blue and a red goes to one cell, and a blue and a red goes to another cell. And at the end, each one of them divide. This is the meiosis II is similar to mitosis. Okay, that the way chromosomes divide is same as mitosis. And then meiosis 1 is they go through prophase, metaphase, anaphase, telophase. Mitosis 2, same thing. They go through prophase, metaphase, anaphase, telophase. Synapses, homologous chromosomes come together, and that's what synapses and line up side by side at metaphase on the metaphase plate during meiosis 1. Keep the 4 chromatids closed, so DNA does not replicate during interkinesis at the time between the meiosis 1 and meiosis 2. That's right. There is no more chromosome duplication occurs after the S phase of meiosis. Meiosis 2, again, these are all right here, meiosis 1 and meiosis 2. Please look at these diagrams, the pictures of the chromosomes, how chromosomes line up, what color with what color, and so on and so forth. It is important for you guys. They are showing a little bit of crossing over right here. You might be wondering, how come this red chromosome has a blue one? It's called crossing over. So a little bit of this chromosome gets tied up with the other chromosome. It breaks off and attaches here. It's abnormal. It happens all the time. It's not an abnormal process. We will talk about some abnormal process here in a minute. So meiosis and genetic variation, very important. Make sure you read those. Right here, he's going over crossing over and metaphase 1, so on and so forth. He's going over the spermatogenesis and oogenesis. It means formation of the sperm and egg. Genesis, it means formation. Formation of the egg and formation of the sperm. Primary oocytes, spermatocytes, and so on and so forth. Again, I'll let you read all of these. Interesting. So at the beginning, the 2N cells, this is a 2N cell, it does not have flagellum, but undergo meiosis 1 and undergo meiosis 2, and then they start having the flagellum. Female, when you start out, yes, you start out with 1 egg. At the end, there are 4 eggs, but 3 of them are called polar body. They cannot continue on the life right here, polar body. Okay, so, but only 1 egg can become viable and move on into the life. Okay, make sure you read these things. These are significant. Again, I don't want to take up too much of your time listening to these. Comparison of meiosis and absolutely make sure you read it. You know the differences between meiosis and mitosis. When school was in session, that was one of the essay questions. I would ask that. I say, okay, what is the difference between, okay, good. And now you put it in a table, comparing the two, make sure you study those and know them. Chromosomes and inheritance, I guess there are books in here explain how non-disjunctions work. So when you have a pair of autosome carriers alleles for particular traits, so as I said, you have two chromosomes, right? They're homologous to each other. So the definition of allele is alternative gene. Allele, it means alternative gene. So if this gene does not work, I guess that one is coming to work. It doesn't, it's not as simple as that, what I told you. Okay? But you have two, when we study doing, when we get to the genetics chapter, you will see there are two genes. For example, capital A, I might as well say capital A, small a. This is alternative to that one, right? You guys follow capital of cap. This is one allele, this is one allele. So anyhow, we'll talk about it when we will see it. So what is non-disjunction during meiosis 1 and meiosis 2? During meiosis, when homologous chromosomes fail to separate properly, that is called non-disjunction. Okay, during meiosis, when the chromosomes fail to separate properly, it's called non-disjunction. So what happens? There are two scenarios. The 2N cells, as you already know, 2N cells have all 46 chromosomes. When during meiosis, the chromosomes, and you will see a couple of scenarios here in a minute, when the chromosomes do not separate properly, what happens? They might get stuck. The 2N, the diploid cell, might get one extra chromosome as well. That is called trisomy. When they do not get one of the chromosomes, opposite of trisomy is called monosomy. Okay, so that is the egg you took 20 versus 23. Okay, so here you go, the diagram showing you exactly what is happening. That's why I said I like your textbook. And in case of trisomy and non-disjunctions and monosomy and so on and so forth. Okay, abnormal autosomes cause development abnormality, monosomy of all but X chromosomes is fatal. So we'll talk about that too. Among autosomal trisomy, usually trisomy 21 Down syndrome has a chance of survival. Turner syndrome, Down syndrome, all of those are important to know. Somewhere in here it gives you the definition of Jacob syndrome. It's XYY, and then Turner syndrome is missing one X. It's most likely reasons that why a zygote with one X chromosome, Turner syndrome can survive. But anyhow, these are the reasons. A syndrome, you guys, it's a term, a syndrome. I was looking for that term. I guess it's not in there. A syndrome is a collective term for having a few characteristics that are very close to each other. For example, bulky head, or the hands that are, I'm giving you the Down syndrome. The hand, the short statures, eyelids are folded, a flat face. These are some of the syndromes. So if somebody has these characters, they say, okay, they're all look alike. So that would be a Down syndrome trisomy. Okay, so the chromosomal trisomy for Down syndrome chromosome. Here we go. That's what a group of symptoms that always occur together. Okay, and determining factors, changes in sex chromosomes, numbers, and the Turner, as I said, Turner syndrome is XO. It's missing one of the chromosomes. That's why it's XO. Klinefelter syndrome. You have 47 chromosomes, and so 46. And it's XXY, one extra X. Okay. are with them 2 chromosomes and 1 Y chromosomes. So the symptoms, speech and language. So these are the symptoms. That's what I say. So speech and language delays. Yes, you should know that these diseases, there are not too many of them, maybe only 4 or 5 your textbook gave you, and the symptoms, signs and symptoms as well. Okay, Jacobs is XYY, and then it can result from non-disjunctions, and then affected the males are tall, and they have persistent acne and speech and reading problems. So, but they are fertile, and they may have children as well. Some of these symptoms they can be fixed by a hormonal therapy. It is in here. By hormonal therapy, they can be fixed. Environment, radiation also can have an effect on the chromosomes, absolutely. And that's what happens. These are the 4 problems that can happen with chromosomes: deletion, duplication, inversion, and translocation. Okay, so deletion, let's have a picture right here. So A is being simply deleted from this chromosome, so the chromosomes are duplicating. Okay, in here you call it duplication, so you have DE and DE being duplicated. These are all problems. Don't think, oh, duplicate, yeah, your money is duplicated. No, it's not like that. All of these can cause problem. Inversions is supposed to be A, B, C, D, and D and C, they switch their places. So that is inversion. Translocation, it means a segment of a chromosomes from this jumps to here, and that one jumps to here, and that is not crossover. When crossing over already occurred, and now translocation, when the chromosomes flip-flop, and that is a problem. Again, crossing over, as I said, it's normal. Usually does not cause any problem. So again, he is talking about all of these, the inversions and translocations, and deletion is Williams syndrome on chromosome number 7. You saw them in karyotype at the beginning of the chapter. Look at chromosome 7. And most of these, it will tell you what chromosomes there is a problem, the diseases that we study. Okay, these are deletions of Williams, deletions, cry to chat, the baby cries like a cat, that's what cats cry. Anyhow, the glottis, that's where the voice box and larynx makes the noise, so when the baby's crying, it's a translocation. He's giving you a few examples, and here, make sure you know these things. Okay. And that's about it. All right, you guys, that's the end of the material for exam number 4. From next on, everything that I talk about is for final exam.